Number 1. The parasite that may have killed Tyrannosaurus rex. Picture the most feared skull in the history of life on land. The jaws are longer than your arm, lined with teeth the size of bananas, built to crush bone. Now look closer at the lower jaw. Along the bone, there are holes. They are round, smooth at the edges, almost as if something had drilled them cleanly through the living tissue. For years, these holes were a mystery, and the answer that eventually emerged is far stranger than any wound from another dinosaur. This is Sue. Officially, she is specimen FMNHPR2081, the largest and most complete Tyrannosaurus rex ever discovered, about 90% intact. She was found in 1990 near the town of Faith, South Dakota, by the fossil hunter Sue Hendrickson, and she is now the centerpiece of the Field Museum in Chicago, which bought her at auction in 1997 for about $8.36 million, the highest price ever paid for a fossil at the time. For decades, the holes in Sue's jaw were blamed on the obvious suspects. Perhaps they were bite marks from another tyrannosaur. Perhaps they were osteomyelitis, a bacterial infection of the bone. But in 2009, a team led by Ewan Wolfe, along with Stephen Salisbury, John Horner, and David Verricchio, published a very different idea in the journal PLOS One. They proposed that the killer was not a rival predator at all. It was a single-celled parasite. The clue was in the shape. The lesions in Sue's jaw look almost exactly like the damage caused by a disease seen in modern birds called trichomoniasis. Falconers know it as flounce. Pigeon keepers call it canker. It is caused by a microscopic flagellated protozoan named Trichomonas gallinae, an organism so small you would need a microscope to see it. In birds today, it colonizes the mouth and throat, producing thick, cheesy lesions that can eventually block the airway and the gullet entirely. Here's why the idea is so plausible. Birds are living dinosaurs, the direct descendants of small meat-eating theropods. A parasite that lives comfortably in birds today could very plausibly have infected their close relatives in the Cretaceous, and the researchers did not stop at Sue. They examined at least 10 tyrannosaur specimens, and several of them carried the same rounded lesions in the same part of the jaw. So how would a microbe bring down a 9-ton predator? Not by eating the bone. The proposed mechanism is quieter and crueler. As the infection advanced, the lesions in the throat would grow until swallowing became painful, then difficult, then impossible. The most powerful hunter of its age would slowly starve, unable to eat the prey it could still catch. Sue lived to around 28 years old, remarkably old for a giant theropod, which is exactly the sort of long life in which a chronic infection has time to take hold. How did the parasite spread? The team suggested a few routes. Tyrannosaurs are known to have bitten each other in the face, and healed facial bite wounds appear across many specimens, evidence of combat or courtship. Passing saliva mouth to mouth could transmit the disease. So could cannibalism or feeding on already infected prey. Not everyone is convinced. The mainstream counter-argument holds that the holes are simply bite injuries or bacterial osteomyelitis, and a further view suggests a fungal infection could produce similar damage. Critics point out, fairly, that the parasite itself could never fossilize, so the whole case rests on the pattern of the bone. Supporters answer that no bite pattern cleanly explains smooth, rounded holes appearing in the same places across different animals. Whatever the truth, the image lingers. The tyrant king of the Cretaceous, perhaps undone by something too small to see. And if the largest killer that ever walked was so vulnerable, what was happening in the water, in the mud, and inside the smallest bodies of the ancient world? Number 2. The first parasite ever found. Travel back 512 million years to a warm, shallow sea long before anything crawled onto land. The seafloor is dotted with small shelled creatures filtering food from the water, and already, at this impossibly early date, something has learned to cheat. Something is stealing. In 2020, a team led by Zhifei Zhang published a discovery in the journal Nature Communications that reset the clock on parasitism itself. Working with fossils from the Guanshan biota in Yunnan, China, dated to about 512 million years old, they studied a small brachiopod, a lampshell named Neobolus wulongkingensis. A brachiopod looks a little like a clam, a soft animal inside two protective shells, feeding by drawing a current of water through its body and filtering out particles of food. Attached to these shells were tiny tube-dwelling worms. At first glance, a hitchhiker riding on a shell need not be a parasite. It could be harmless. What made this case different was geometry. The worm's tubes were consistently oriented with their open ends pointing toward the host's feeding current, positioned precisely to intercept the food the brachiopod had worked to gather. This is theft, a lifestyle biologists call kleptoparasitism, meaning the parasite steals food rather than eating the host's body. But orientation alone is an interpretation. The team needed proof of harm, because the strict definition of a parasite is an organism that lives at a cost to its host. So they measured. Examining more than 100 specimens, they found a clear pattern. Brachiopods carrying more worms were consistently smaller. The worms were stealing enough food to stunt their host's growth. That measurable cost is the signature of true parasitism, and it separates this relationship from mere commensalism, where a passenger neither helps nor harms. At 512 million years old, this is the oldest known parasite-host relationship in the entire fossil record, with the harm actually demonstrated rather than assumed. And the timing's astonishing. This falls within the Cambrian explosion, the burst of evolution when most major animal body plans first appeared. It means that parasitism, one of the most common ways of living on Earth, was up and running almost the instant complex animal ecosystems existed. Parasitism may be exactly as old as predation, both born in the same ancient sea. We still do not know what these worms actually were, because only their hard mineral tubes survived, not their soft bodies. Skeptics rightly caution that reading behavior from fossils is difficult. But the two-part argument, the tube orientation combined with the measured growth cost, is far stronger than either line alone. The lesson is chilling in its simplicity. From the very dawn of animal life, no sooner had bodies appeared than other organisms evolved to live off them. And that pressure never stopped. It only grew more inventive, as the next fossil shows in a way you can almost watch happen. Number 3, caught in the act 425 million years ago. Almost every fossil of a parasite is a clue left behind, a hole, an egg, a swelling, from which we must infer the crime. Only very rarely does deep time hand us the criminal still gripping the victim. In 2015, that is exactly what a team led by David Siviter, with Derek Siviter, Derek Briggs, and David Legg, described in the journal Current Biology. The fossil comes from the Herefordshire Lagerstätte in England, a remarkable deposit formed from volcanic ash on an ancient seafloor about 425 million years ago in the Silurian period. The animal they named is Invivida piratica. The names are not subtle. Invivida means the intruder and piratica means piracy. This is a parasite named for theft, and it was preserved locked onto its host. Invivida is a pentastomid, commonly called a tongueworm. These are strange creatures, parasitic relatives of the arthropods, the great group that includes crabs and insects. A pentastomid has a segmented, limbless, worm-like body, and these ancient ones were tiny, only about 1 to 4 millimeters long. Today there are around 140 species of tongueworm, and almost all of them live inside the lungs and nasal passages of reptiles, especially snakes and crocodiles. That is what makes this fossil so important. The Herefordshire specimens were found attached to the outside of an ostracod, a small crustacean that lives inside a hinged bean-shaped shell. Some of the parasites were positioned near the host's eggs, suggesting they were feeding on the developing brood. Before this discovery, fossil tongueworms were known almost only as scattered, isolated juveniles. Never before had one been found together with its host, caught mid-attack. How do you study a soft creature barely a few millimeters long, sealed inside solid rock? The team used a painstaking method. They ground the fossil away in extremely thin layers, photographing the exposed surface at every step, then stacked the images to build a detailed three-dimensional digital model. The catch is brutal. The technique physically destroys the fossil as it records it. The digital reconstruction becomes the only surviving specimen. The implications reach across 250 million years. It shows that tongueworms were parasitizing marine invertebrates in the Silurian long before there were any reptiles to infest. Their later move into the lungs of snakes and crocodiles was a change of address that came much later. Their specialized parasitic anatomy, the clinging body, the simplified form, was already perfected 425 million years ago. And if a parasite this specialized already existed in the Silurian seas, imagine what was waiting for the giants that would soon rule the land. Number 4. Giant fleas that fed on dinosaurs. You know the modern flea as an almost invisible speck, a dark dot that vanishes the moment you try to pin it down. Now magnify that image until the flea is nearly the length of your thumbnail, arm it with legs like grappling hooks and a mouth part like a serrated blade, and set it loose on the skin of a dinosaur. That animal really existed. In 2012, a team led by Ding Huang described these creatures in the journal Nature, giving them names like Pseudopulex jurassicus and Pseudopulex magnus. They come from two of China's richest fossil beds, the Middle Jurassic of Inner Mongolia and the early Cretaceous Yixian Formation of Liaoning, spanning roughly 165 to 125 million years ago. The largest reached about 2 centimeters in body length, 5 to 10 times the size of the fleas that trouble a dog today. These were not simply big fleas, they were built differently. A modern flea is flattened side to side, a shape that lets it slip between hairs, and it is a champion jumper. These ancient giants were flattened the other way, top to bottom, and they could not jump at all. Instead, they had long legs tipped with strong claws, ideal for clinging to a host and holding on, and their mouthparts were extraordinary, long, rigid, and edged like a saw, clearly built to pierce something tough. Something tough is the key. The likely hosts were the feathered dinosaurs and early mammals that shared these Mesozoic forests, animals with thick hide or dense hair-like proto-feathers. A delicate mouthpart would be useless against a dinosaur. These blades were made to drive through it and reach the blood beneath. Because the insects could not leap, they most likely crawled aboard their hosts and simply held fast. Where exactly these creatures sit on the family tree is debated. Some researchers class them as true fleas in the group called Siphonaptera. Others place them in an extinct-related lineage, an early experiment in blood feeding that predates the modern flea body plan. A related giant parasite described under the name Saurophthyrus may even have targeted the flying pterosaurs. The Yixian formation that preserved them is world famous for its feathered dinosaurs, ancient birds, mammals, and insects, all entombed in fine lake sediment by volcanic activity, flattened into exquisite detail. There's a darker thread here, too. Modern fleas are notorious disease carriers, most infamously as the vectors of plague. Whether these Mesozoic giants spread disease among dinosaurs is pure speculation, because no ancient pathogen has been recovered from them. But the possibility that dinosaurs itched, bled, and perhaps sickened from the bites of fleas, the size of your thumbnail changes how you picture them. And if parasites were riding on the outside of dinosaurs, others had already learned a far more disturbing trick, growing on the inside. Number 5. The Chestburster, wasps, hidden inside fossil pupae. Imagine a coffin the size of a grain of rice. From the outside it looks ordinary, just the hardened case of a fly waiting to transform. But inside, where the fly should be, something else has been growing. Something that ate the fly alive and took its place. For tens of millions of years, this scene stayed hidden, until a machine let scientists see straight through the stone. In 2018, a team led by Thomas van de Kamp published a study in Nature Communications that reads like a horror film written in fossils. They used a synchrotron, a particle accelerator that produces intense X-rays, to perform micro-CT scans, essentially a hospital CT scan at microscopic resolution on fossil fly pupae. The specimens came from museum collections, from deposits in France dated to roughly 30 to 40 million years ago. They scanned an enormous number of pupae, nearly 1,510 of them. And inside 55 of those little cases, they found something that was not a fly. They found wasps. Parasitic wasps had laid their eggs in or on the developing flies, and the wasp larvae had devoured their hosts from the inside, growing into fully formed adults sealed within the fly's own coffin. The team named several new species, and one of them they called Xenomorphia, a direct nod to the creature from the film Alien, because the life cycle is almost exactly the same, an organism developing inside a living host and emerging by killing it. These killers are called parasitoids, and they differ from ordinary parasites in one grim detail. A parasite usually keeps its host alive. A parasitoid almost always kills it. The scanning caught the whole drama in freeze frame. Some wasps were preserved mid-development, still growing. Others were fully formed adults that had died before chewing their way out of the case. The mineral that had slowly filled each pupa preserved the internal anatomy in perfect 3 dimensions, completely invisible from the outside. This is one of the largest studies of ancient parasitism ever attempted, because it was not a single lucky fossil, but a statistical survey of thousands. 55 parasitized cases out of about 1,510 even lets researchers estimate how common this parasitism was in that ancient ecosystem, and the presence of several different wasp species means a whole community of parasitoids was attacking the same host flies. Parasitoid wasps today are among the most diverse groups of animals alive, with tens of thousands of species. And here, the deep past reaches straight into today's headlines. The threat spreading through the Americas right now, the New World screwworm, is a fly whose larvae eat the living flesh of animals from the inside of a wound. It is a different creature from these wasps, but the strategy, growing by consuming a living body from within, is the very same one these fossils show was already perfected 40 million years ago. Nature discovered that horror long before it reached a modern ranch. Number 6, the Body-Snatchers preserved in stone. Some parasites do not simply feed on their host, they take it over, they move in, shut down the host's ability to reproduce, and turn the entire animal into a life support system that exists only to feed the invader. And this is called parasitic castration, and it is one of the most unsettling strategies in all of biology. Astonishingly, we can see its fingerprints in fossils more than 100 million years old. The culprits belong to a group of parasitic isopods called bopparids. Isopods are crustaceans, relatives of the woodlouse, and bopparids specialize in infesting other crustaceans, lodging beneath the shell, often in the gill chamber, and draining nutrients from the host. Today, they are a genuine problem in shrimp and crab fisheries. As the parasite settles in, it frequently sterilizes its host and produces a distinctive bulge, a swelling in the shell where the parasite sits. That swelling is the key because while the soft parasite itself rots away and never fossilizes, the deformation it carves into the host's hard shell can last for ages. In 2014, a study led by Audio Klompmaker documented these swellings as trace fossils, evidence of behavior rather than a body, and gave them a formal name, the Ichnotaxon Conthyloma crusta. A trace fossil records what an organism did, like a footprint or a burrow, and here, the trace is the scar of an infestation. The record runs deep. These bopparid swellings appear in fossil crabs, lobsters, and shrimp going back to the Jurassic, roughly 150 million years ago or more. They show up as a rounded bulge, usually on just one side of the animal's carapace, exactly matching the position of a single lodged parasite. That one-sided placement is the crucial detail that separates a parasite swelling from a random injury or a disease that would affect the whole shell. Klompmaker and colleagues surveyed large museum collections to trace how common these infestations were across geological time, and found that their frequency roughly tracked the great diversification of crabs and their relatives through the age of dinosaurs and beyond. This is the same broad strategy used by the modern parasite Saculina, a barnacle so transformed by parasitism that it no longer looks like a barnacle at all, and which castrates crabs and hijacks their behavior, though it leaves a different kind of trace. Because the host survives sometimes for a long time, these fossils record not a quick killing, but a chronic drawn-out relationship, an animal kept alive and sterile so that its body could serve the parasite. That such a specific sinister strategy has persisted, essentially unchanged for well over 100 million years, tells you how successful it is. And if parasites could reshape a body from within a shell, others could freeze the exact instant of a killing in a drop of golden resin. Number 7. The worm bursting from its host, frozen in amber. A drop of tree resin falls, slow and thick, onto a struggling insect. In that same instant, something is erupting out of the insect's body, a long, thin worm forcing its way into the open. Then the resin closes over both of them and holds that single second of violence for 99 million years. That is not imagination. That is a real fossil. Burmese amber, from the Hukong Valley of northern Myanmar, is roughly 99 million years old, dating to the middle of the Cretaceous. Unlike most fossils, which are flattened and mineralized, amber preserves organisms in 3 dimensions, with soft bodies, fine hairs, and delicate limbs intact. It has yielded insects, spiders, and even fragments of feathered dinosaurs. And crucially, because resin traps a creature almost instantly, it can capture behavior, a single frozen moment of something happening. The scientist most associated with these discoveries is George Poinar Jr. of Oregon State University, who has spent a career finding parasitism sealed in ancient resin. Among the most dramatic are myrmidid nematodes, parasitic roundworms that develop inside insect hosts and then kill them as they emerge. In amber, these worms have been found in the very act of exiting their hosts, coiling out of the bodies of midges and other small insects. Because a myrmidid's emergence normally kills the host, these are, in effect, fossilized moments of death. Poinar has documented many other parasites in amber too, including mites clinging to their hosts and biting insects preserved with the microscopic organisms they carried. Burmese amber has even produced ticks tangled in the feathers of dinosaurs, direct evidence of external parasites feeding on them. The clarity of the resin allows all of this to be studied under a microscope in fine anatomical detail, without destroying the specimen, quite unlike the grind and photograph method used on the Silurian tongue worm. There is a serious shadow over this science. Much Burmese amber comes from a region torn by armed conflict, which has prompted difficult ethical debate among paleontologists about whether and how it should be studied and collected. That conversation continues, and it matters. What the amber makes undeniable is that internal worm parasitism, one of the most common lifestyles on Earth today, was already fully operational in the Cretaceous, exploiting the same kinds of insect hosts we see it used now. Soft-body worms almost never fossilize in ordinary sediment, so without amber, this history would be nearly invisible. Between the tick in the feather, the worm bursting from the midge, and the parasites and the biting flies, amber shows creatures being exploited in the air, in the water, and inside their own bodies, all at the same moment in deep time. And the inside of the body is where the next parasite left its mark, in a place you would never think to look. Number 8. The oldest tapeworm, hidden in fossilized dung. Some of the most important parasites in history left no body, no shell, and no bones. They lived coiled in the guts of other animals, soft and boneless, and when their host died, they simply decayed. So how could we ever find one from before the dinosaurs? The answer is as unglamorous as it is brilliant. You look inside fossilized feces. In 2013, a team led by Paula Dencian-Diaz published a discovery in the journal Outbreak 1 that pushed the history of tapeworms deep into the ancient past. They were studying a coprolite, a piece of fossilized dung from the Rio do Rosto Formation in southern Brazil, dated to the middle to late Permian, around 270 million years ago. Based on its shape and the fossils found around it, the dung was attributed to a shark. When they examined it closely, using thin sections of the rock and a scanning electron microscope, they found something remarkable sealed inside. A tight cluster of at least 93 tiny eggs. The eggs were small and oval, measuring about 145 to 155 micrometers, far thinner than a human hair, and some of them preserved internal structures. One appeared to show a possible larval hooklet, the kind of feature seen in the infective stage of a tapeworm. The eggs were packed together in the way a tapeworm segment releases them. In a dense little parcel, this is why coprolites are so precious. A tapeworm's body could never survive, but its eggs have tough, durable shells, and when they are sealed inside a protective lump of dung, they can endure for hundreds of millions of years. This single find pushed the confident fossil record of tapeworms back roughly 200 million years earlier than the previous solid evidence, showing that internal gut parasitism of vertebrates was already established long before the first dinosaur walked. The timing carries extra weight. 270 million years ago is deep in the Permian, and the period ended in the largest mass extinction in Earth's history. This tapeworm lineage was infecting sharks before that catastrophe struck, part of a story of internal parasites that stretches from the Paleozoic seas all the way to the modern day, when tapeworms still infect fish, birds, mammals, and humans. The mainstream view treats identifications like this with appropriate caution, because eggs from different parasite groups can look alike. The authors argued that the size, shape, and tight clustering of these eggs point specifically to a tapeworm-like flatworm, and the case has stood as one of the oldest direct fossils of a specific internal parasite of vertebrates. It also proved that fossil dung is one of the richest and most underused archives of ancient disease we have. But not all parasites hide in the distant past of sharks and dinosaurs. Some of the worst crawled straight into human bodies, and we can still find them there. Number 9. The fiery serpent, drawn from an Egyptian mummy. Now the story comes home into the human body. Imagine a worm, thin as a strand of spaghetti, but as long as your arm, living quietly inside a person's leg for a year. Then one day, it begins to move toward the surface, and it emerges, slowly, through a blister of burning skin. The ancients had a name for the agony of it. They called it the fiery serpent. The creature is real, and it still exists. It is Dracunculus medinensis, the guinea worm, a parasitic nematode. The female can grow up to about 1 meter long inside the human body before she emerges. People become infected by drinking water that contains copepods, tiny water fleas carrying the worm's larvae. Inside the body, the worm matures, migrates through the connective tissue, and eventually pushes out through the skin, usually of the lower leg or foot, in a painful burning blister. The traditional treatment is thousands of years old and is still used today. When the worm's head emerges, you wrap it around a small stick and turn the stick a little each day, drawing the worm out slowly over days or weeks because pulling too hard will snap it and cause serious infection. Some scholars have even suggested that the ancient medical symbol of a serpent wound around a rod may be connected to this very practice, though that link is debated. We know this parasite plagued the ancient world because it left physical traces. A calcified guinea worm, a worm that mineralized within the tissues, has been reported preserved in an Egyptian mummy. And the written record is just as striking. The Ebers Papyrus, an Egyptian medical text dated to around 1550 BCE, is widely interpreted as describing guinea worm and its extraction, and the fiery serpent of ancient Near Eastern texts is often identified by scholars as this very disease. This kind of research, hunting parasites in mummies, coprolites, and the soil of ancient latrines, is a whole field called paleoparasitology. Here, the story turns from horror to hope. Guinea worm has been the target of a global eradication campaign led since the 1980s by the Carter Center and its partners. The results are among the great triumphs of public health. Cases have fallen from an estimated 3.5 million a year in the mid-1980s to only a low double-digit number of human cases in recent years. Guinea worm is now on track to become only the second human disease ever wiped out, after smallpox, and the very first parasitic one. What makes it extraordinary is how it is being beaten. There's no vaccine and no drug. The weapons are filtering drinking water and containing each emerging worm, so it cannot reinfect the water supply. An ancient scourge defeated by simple, patient means. The final push has been slowed by an unexpected twist, guinea worm infections turning up in dogs, but the end is in sight. From a worm calcified in a mummy to a disease almost erased from the earth, this single parasite ties the deep past directly to the living present. And one last parasite turns that same trick, using a worm to rewrite not just medicine, but the entire story of how people reach an entire hemisphere. Number 10. The parasite that rewrote the peopling of the Americas. A gutworm should be a footnote in the story of a mummy, not a challenge to the settlement of two continents. Yet a humble hookworm, found inside ancient South American remains, has forced archaeologists into one of their most stubborn arguments, and it rhymes, uncomfortably, with the Outbreak crossing toward the United States today. Hookworms are blood-feeding intestinal parasites. The two species that infect humans are Ancylostoma duodenale and Necator americanus, and that second name is worth pausing on, because Necator americanus literally means the American killer, a reflection of how common it once was across the American South. Hookworm larvae live in warm, moist soil, and they infect people by penetrating the skin directly, usually through the bare feet of someone walking across contaminated ground. Researchers including Adelia Cianto, Carl Reinhard, Luis Fernando Ferreira, and Adauto Araujo have documented hookworm and other parasites in ancient remains across South America. Their eggs and larvae have turned up in pre-Columbian mummies and coprolites in Brazil, Peru, and Chile, some of them thousands of years old. One frequently cited coprolite from Brazil yielded hookworm evidence dated to roughly 7,200 years ago and possibly older. Araújo and Ferreira were pioneers of paleoparasitology, building much of the field at Brazil's Fiocruz Institute around exactly this kind of work. And that is where the trouble begins because of one simple biological fact. Hookworm larvae cannot survive prolonged freezing. The standard model of how humans first reached the Americas has them crossing from Asia through Beringia, the land bridge exposed during the last ice age around 15,000 or more years ago, through bitterly cold conditions. A hookworm riding in the gut of a human walking that frozen route should have died long before reaching the other side. So how did a warm climate parasite end up deep in ancient South America? This is a genuine, unresolved scientific puzzle, and several explanations compete. One holds that early people took a coastal route, moving along milder shorelines rather than the frozen interior. Another suggests warmer inland corridors, or that people arrived earlier than the classic model allows. Some researchers have even floated lower latitude or transoceanic entry to explain how warm-adapted parasites made the journey, though these ideas are contested and remain outside the mainstream. What everyone agrees on is that parasite eggs are powerful evidence. They are durable, and they are specific to particular species, which makes them precise markers of where humans went and how they lived. The presence of the human-specific parasites also tells us about population density and sanitation in ancient settlements. The hookworm puzzle is still not solved, with genetic, archaeological, and parasitological evidence still being fitted together. Modern hookworm, meanwhile, still infects hundreds of millions of people and remains a major cause of anemia in children. A tiny worm requiring only warmth to survive has become a clue about the deepest chapter of human prehistory, proof that parasites are not just agents of disease, but archives of history written inside the body. Step back from the individual horrors and a single pattern comes into focus, stretching across more than 500 million years. The same lifestyle appears again and again in every environment, on every kind of host. Live off another body. Take what you need, give nothing back. Follow the thread. The Cambrian tube worms at 512 million years show parasitism present at the very dawn of complex animals. The Silurian tongue worms at 425 million years show the clinging, simplified body of a specialist already refined. The Permian tapeworm at 270 million years shows internal gut parasitism established before the dinosaurs. Then come the Mesozoic fleas riding on dinosaurs, the amber worms bursting from Cretaceous insects, and the isopods castrating crabs, followed by the parasitoid wasps of the Paleogene killing flies from the inside, and finally the human parasites, the guinea worm and the hookworm, showing the same ancient pattern reaching all the way to us. This is not a series of freak events. It is the norm. Parasites likely outnumber free-living species on Earth, and essentially every major group of animals has, over time, acquired its own suite of parasites tuned to exploit it. Each pairing drives a coevolutionary arms race, the host evolving defenses, the parasite evolving ways around them, over and over across geological time. Parasitism has arisen independently many times across the tree of life, because in evolutionary terms, it is a cheap and effective strategy. Why build your own body's resources when you can borrow another's? It is worth remembering that the fossils we have represent only a floor, never a ceiling. Soft-bodied parasites almost never preserve, so the true history is far richer than the scraps we can see. Those scraps come to us in three recurring ways: as body fossils, as trace fossils, like shell swellings, and as organisms sealed in amber or eggs in coprolites, each mode capturing a different slice of behavior. And the oldest evidence keeps getting older as our methods improve. Mass extinctions have repeatedly wiped the slate of host communities clean, yet the parasitic strategy itself always survives and returns, because wherever there is a living body, something evolves to exploit it. The obvious question, after all of this, is how anyone can possibly study creatures that were soft, tiny, and boneless, and that died hundreds of millions of years ago. The answer is a kind of detective work, and once you see the tricks, you start to see the ancient world differently. The first tool is fossilized dung. Coprolites act like sealed time capsules, protecting the durable eggs of gut parasites long after the worms themselves have vanished. Scientists slice them into paper-thin sections and examine them under scanning electron microscopes, revealing eggs measured in micrometers. The second tool is the trace fossil, the mark a parasite leaves on something hard. A one-sided swelling in a crab's shell records an isopod that has long since rotted away, and researchers give these traces their own formal names, like Canthaloma crusta, so they can be studied systematically. The third tool is amber, which entombs whole soft bodies in 3 dimensions and can freeze a single instant of behavior, like a worm mid-emergence. To look inside solid specimens, scientists now use synchrotron X-ray micro-CT scanning, which sees through stone without harming the fossil, the very technique that revealed the wasps hidden inside fly pupae. When a fossil is too small and delicate for that, they sometimes use the destructive grind and photograph method, sacrificing the specimen to build a perfect digital model. On bone, they read disease directly from the pattern of damage, the way the lesions in Sue's jaw were interpreted. There is even a dedicated field for the human side of this story, paleoparasitology, which examines mummies, ancient latrine soils, and preserved gut contents to trace disease through human history. Across all of it, the same principles apply. Egg shape and size help identify a parasite group. Sampling thousands of specimens reveals ancient infection rates rather than single anecdotes. Comparison with living relatives keeps interpretations grounded, and molecular clocks which estimate divergence times from genetics provide an independent check on the fossils. No single method is foolproof, and each carries the risk of a false reading, which is exactly why the strongest discoveries combine several independent lines of evidence at once. Some of the best finds are not dug from the ground at all, but rediscovered in museum drawers when old specimens are scanned with new machines. For all of this, the honest picture is full of open questions, and that uncertainty is part of what makes this subject so alive. Take the very first case. Whether a Trichomonas-like parasite truly killed Tyrannosaurus rex is still unproven, and always may be because the microbe itself can never be recovered. The identity of the Cambrian tube worms remains unknown. The exact origins and earliest hosts of the tongue worms are still debated. The list goes on. Whether the giant Mesozoic fleas actually transmitted disease among dinosaurs is pure speculation. How representative that 55 out of 1,510 parasitism rate is for the ancient world is uncertain. The full diversity of soft-bodied parasites in the Paleozoic seas is almost entirely hidden from us. Egg identifications inside coprolites can be genuinely ambiguous, and different parasite groups can leave eggs that look frustratingly alike. Above all, the route by which worm-loving hookworm reached the ancient Americas is still unresolved, one of the most stubborn puzzles in the field. Bigger questions loom behind the specific ones. We do not fully understand how parasites survive mass extinctions when so many of their hosts vanish, or whether parasitism as a whole spikes or crashes across those catastrophes. The earliest true tapeworm ancestor may be far older than 270 million years. Molecular dates and fossil dates for parasite lineages often disagree, sometimes sharply. The dating and ethics of Burmese amber complicate an entire archive of Cretaceous life. Many supposed traces of parasitism might have innocent explanations, and the behavior behind every fossil is inferred, never actually witnessed. Ancient parasite D, Na, is rare and extremely hard to recover, and the interplay between climate and the spread of parasites in deep time is barely studied. Whether parasites help to drive the evolution of their hosts, including our own species, is a live debate. And every time a new imaging technique arrives, another oldest record falls. In this field, each answer tends to raise a harder question. All of this deep time horror sharpens the way we should look at the present and at the outbreak in the headlines right now. The New World screwworm, Cochleomiae hominivorax, is a fly whose females lay their eggs in the open wounds of living animals. What makes it so feared is that, unlike most maggots, which feed only on dead tissue, screwworm larvae burrow into and consume living flesh, which can be lethal to livestock, wildlife, and occasionally people. The screwworm was eliminated from the United States decades ago through one of the cleverest tools in pest control, the sterile insect technique, in which vast numbers of sterilized male flies are released so that wild females mate without producing offspring, collapsing the population. For years, a barrier maintained around Panama kept the fly bottled up in South America. The renewed alarm comes from its recent spread north through Central America and into Mexico, moving back toward North America and triggering restrictions on livestock movement along the way. It is, in a sense, a modern echo of the parasitoid wasps preserved in amber, a creature that lives by consuming a body from within. The deeper record puts this in perspective without dismissing it. A warming climate can widen the range of many parasites and the insects that carry them. And thawing permafrost and ancient remains have raised concern about long-dormant pathogens. Ticks and fleas remain major disease vectors today, exactly as the amber fossils show they were in the Cretaceous. Globalized trade and travel now move parasites around the planet faster than at any time in history. Yet the same record also offers reassurance. Guinea worm stands on the edge of eradication, proof that even an ancient scourge can be beaten with surveillance and simple interventions like water filters, while hookworm, still infecting hundreds of millions, reminds us how much work remains. What the fossils teach most clearly is that parasites always adapt to new hosts and new climates. Today's outbreaks are not a break from nature. They are the newest entries in a pattern that is more than 500 million years old. And the screwworm, for all the disruption it causes, is a small thing beside the sheer depth and variety of the parasites that came before it. What all of this reveals is that parasitism is not some rare aberration of nature, but one of its oldest and most successful ways of being alive. From a worm stealing food from a shellfish in a Cambrian sea, to a fly threatening cattle on a modern ranch, the same quiet strategy has run unbroken through every age of life on Earth. The fossils are snapshots of a single, continuous conversation between hosts and the things that live off them, a conversation that has never once fallen silent. And the most humbling lesson in it is that no host, however mighty, has ever been exempt. The tyrant king of the Cretaceous may have been brought down by a creature too small to see. Human beings, the species that split the atom, still wind meter-long worms out of their own skin on a stick, exactly as their ancestors did thousands of years ago. Remember, too, that what we can see is only the fraction that could fossilize. The true history of parasitism is vaster and stranger than the fragments preserved in stone, dung, and amber, and every new tool pushes that history deeper and reveals it in sharper detail. These parasites are, in the end, archives, carrying the record of their hosts inside living bodies across unimaginable spans of time. The outbreak spreading across the Americas today is real, and it matters, but it is a single chapter in a book with no clear beginning. Seen against that depth, fear gives way to a kind of awe. The smallest organisms have shaped the largest stories, and what looks at first like pure horror is also evolution's oldest and most enduring form of ingenuity. Somewhere, in a museum drawer or a fresh block of amber, the next oldest parasite is already waiting to be found.